Okay. Good morning, students. Good morning, all. And today we will be discussing the classification of enzymes as well as the effect of the different factors on enzymatic activity. So this will be our today's discussion. And yesterday regarding the enzyme we have done the lock and key model, then we have done the activation energy. So before starting of today's class, once again, what is activation energy? Let me see regarding your progress of yesterday's class. What is activation energy? Quickly send me the definition in the chat box and then I will progress with today's class. Once again, what is activation energy? Very good, quick answer, Megha. Shrishti. Correct. Very good. The minimum energy required to start a reaction. Okay. Very good, quick answer. Okay. Very good. Okay. So, yes, fine. Okay. Yesterday worksheet we completed in class only. Yesterday worksheet we completed in class only. Okay, now let me move to the topic classification of enzyme. Yes, my slide is visible. My slide is visible to all. Okay, fine. Now students, here you can see the category of enzyme. Okay, I got the feedback. Yes, it is visible. Now, see, numberings are given, an enzyme category is given, and the function is also given in this table. First, the oxido reductases. The category is oxido reductases. From the name itself, it should be clear to you all that it catalyzes oxidation reduction reaction. Students, can you give any example of an oxidation reduction reaction in the living system? As because you have done this portion previously also, that's why I'm asking you. Can you give any example? Can you give any example? Okay, Anshu. Okay. Yes, Mihir, correct. Yes. Okay. Okay. Yes, correct, students. This is the classification, or we can say the first category of the enzyme, which is the oxido reductases. It catalyzes oxidation reduction reaction. One example of oxido reductase, cytochrome oxidase. Students, we have done respiration. Can you tell me what is the location or where this enzyme cytochrome oxidase is present? Where this enzyme cytochrome oxidase is present? In which part it is present? Cytochrome oxidase? Very good. Yes, I got the quick response. Correct. Yes. Correct. In mitochondria. Very good. Now, the second category is transferase. Yes, Pallavi. Yes, I have seen the answer. Yes, in the mitochondria. So, cytochrome oxidase is the oxido reductases group of enzyme which do or brings about the oxidation reduction reaction or catalyzes the oxidation reduction reaction. Second is the transferase. From the word transferase only, it is clear to us, or it should be clear to us, transfer of group. Transfer of group. Glutamate pyruvate. Glutamate pyruvate is a very good example of the in category transferase. Glutamate pyruvate. Can you tell me where this enzyme is present? Glutamate pyruvate transaminase. Particularly if we will consider the function of glutamate pyruvate transaminase, so in which organ of the body its function, in which part of the body it is functioning? Definitely the analysis come with the serum analysis, but in which part? In which part it is present? Liver. Very good, Pallavi. Very good, Pallavi. That is a liver. As we are saying that it is transferring the group, so it is transferring the group from glutamate to pyruvate. Yes, in the liver. Correct. So now we will come to the third mode or the third category, hydrolases. Hydrolases, that means it will do hydrolysis. This is a very common chemical reaction in the body. Students, can you give example of any hydrolysis reaction in the body? Can you give any example of the hydrolysis reaction in the body? You can think for the you can think for the digestion process. Definitely for the digestion process you can think. Okay, okay, Anjali. Yes, if you will start with this amylase, hydrolyzes starch. Yes, correct. Now the next group is lysases. That means addition or removal of groups to form double bond. Okay, so this lysases example we have done in the respiration 1 cycle. Can you remember the cycle? Can you remember any of the cycle? No, no, Mihir, no. See students, I am since many of the enzymes we have studied, and now when we are doing the category, so the function I am asking you all, because we have done many of the biochemical pathway. We did a number of biochemical pathway or the metabolic pathway. That's why okay, Pragya, hydrolases, okay, brings about hydrolysis. For example, amylase hydrolysis starch. Okay, amylase hydrolysis starch. Starch is amylose, so it brings about the hydrolysis of the starch. Is it okay, Pragya? Pragya, is it okay? Okay. Just a second. Okay, fine. Just I am checking once the participation list. Okay, fine. So it is the lysases. Lysases, I am asking you again, lysases, can you remember any of the, in the TCA cycle, or we can say that have you heard about the name of the enzyme which we have done previously? Carboxylase. Carboxylase. Carboxylase, students, carboxylase. In which of the pathway we have studied carboxylase? This is very, very important, because I told you in the chapter of photosynthesis, in the chapter of respiration, there are metabolic pathway, and metabolic pathway definitely it's very important. Okay, Amrita, yes. Even if the very first metabolic pathway you can remember, yes, Pragya, very good. This is the first metabolic pathway which we studied in class 11, that is a C3 pathway, RUBP carboxylase. So, what is the other name of C3 pathway? The most popular, we popularly remember C3 pathway as, Pragya, spelling is wrong. Correct, Ruchi? Calvin, yes, correct now, correct, yes, students, correct. Calvin cycle. So there you have seen that RUBP carboxylase incorporate atmospheric carbon dioxide, that means addition of group, and then it forms, what is the name of the first product which is a 3-carbon compound? What is the name of the first product which is a 3-carbon compound in Calvin cycle, for which the name C3 is given to it? Very good, PGA, phosphoglyceric acid. 3-PGA, phosphoglyceric, yes, or we can say phosphoglycerate 2, correct, very good. So, correct, yes, all the students except Amrita. Amrita, I think you got confused. You have written pyruvate. Pyruvate is not the first stable product of C3 pathway. The C3 pathway is called C3 pathway because the first stable product, PGA, phosphoglyceric acid, is a 3-carbon compound. Okay? Is it okay, Amrita? Is it okay, Amrita? Okay, fine. Now, the next group of the enzyme is isomerase. That means isomerization. It brings about isomerization. So, isomerase in one very important biochemical pathway of respiration chapter you have done. Can you name the pathway? See, all the examples I am bringing, students, from what we have studied, then it will be easier for you to remember also. Instead of giving new examples, I am giving you the examples which we have already done, and you must be knowing that this biomolecule is the portion given in the 3rd unit, okay? And this is a rediscussion of the chapter, and after that we did the chapter like photosynthesis, respiration. Let me see your answer once. Okay. Okay. If you can remember the glycolysis, glucose 6-phosphate get converted into fructose 6-phosphate. Okay. If you can remember the step of glycolysis, glucose 6-phosphate converted into fructose 6-phosphate in presence of the enzyme in presence of the enzyme phosphohexose. Okay. So it is the isomerization reaction. Okay, is it clear to all? Is it clear to all? And finally, the last group is the ligase. Okay, ligase where, okay, ligase where the union of the two molecules will be taking place. For example, here again you can correlate with the example which we have done in the C3 pathway, where RUBP carboxylase catalyzed joining of RUBP rubulose 1,5 bisphosphate along with carbon dioxide to form the PGA. So whenever the joining of the two molecules is taking place, so we can say that RUBP carboxylase is acting as a carboxylase as well as ligases, because it is joining two molecules. It is joining two molecules. Is it clear to all? So once again, all the enzyme categories: oxido reductase brings about oxidation reduction reaction, transferase transfers the group, hydrolases brings about hydrolysis, lysases removal or addition of group, isomerase brings about isomerization, and ligase ultimately the joining of the two molecules. Is it clear to all? Is it clear to all? Okay, fine. Then rapidly I will ask, or I can say randomly I will ask question from this now. So student, okay, ligase Aditi, I repeat once more as you have said. See, RUBP carboxylase in the C3 cycle, this RUBP carboxylase combines two molecules, RUBP, which is your rubulose 1,5 bisphosphate, along with carbon dioxide, and it ultimately forms the first stable product of the C3 cycle, which is your phosphoglyceric acid, PGA. So this ultimately forms a new compound by the joining of the two molecules. So RUBP carboxylase, which was acting as a carboxylase, also act as a ligases. Is it clear, Aditi? Aditi, is it clear to you now? Okay, fine. Okay. Phosphohexose Pragya, the step of glycolysis where glucose 6-phosphate was converted into fructose 6-phosphate, that was isomerization reaction, phosphohexose. Is it okay, Pragya? Phosphohexose isomerases, the name isomerases is given to it, phosphohexose isomerases. Pragya, is it clear to you now? Okay, fine. So now I will proceed to the next topic, which is the factor affecting the enzyme activity. Before opening the slide, factor affecting the enzyme activity, student, through your chat box, can you recall the factors which affect enzyme activity? Only the name of the factor. How it affects, I don't want now, but what are the factors that affect? Anyone? Okay. You send your reply through chat box. Meanwhile, I am changing my slide. Just a moment. Okay, you send your reply through the chat box. One minute. Okay, okay, doesn't matter. I think there is some error with opening of the file. No problem. Okay, okay, no problem with this. So, okay, factor affecting the enzyme activity, as you have, some of you have given the points also. Let me see. It's temperature, pH, enzyme concentration, substrate concentration. So, okay, fine. Okay, correct. Okay. Now, students, with the temperature, with the increase in the temperature, the enzymatic reaction or the enzymatic speed, the rate of the reaction increases initially, but do it increases with further increase beyond the optimum range? Do it increases permanently beyond the optimum range? I wanted to show you with that graph only. Okay, okay. One thing, let me open the okay uh, NCRT text, NCRT text diagram. I can definitely open with that, and the slide is not opening, but the text definitely. The text image I will share you with you all, so that we can locate there the variation in the, with the help of the graph definitely. Yes, my screen is visible definitely. I think it is clearly visible. Okay, let me come to the content portion. It is visible, yes. I have also seen. Just a moment. Yes, students, yes. I wanted this graph in the slide only, but since that slide is not opening, so I have referred to another image. Here, students, you can see three graphs are plotted. The first one, A, the effect, or we can say the enzymatic activity which will be affected with the change in pH, then again the temperature, and finally the substrate concentration. From the graph, students, you can very easily understand that initially with the increase in the temperature or the pH, the reaction rate accelerates. After reaching to a maximum level, which would be the maximum level? This will be the maximum level for this, and this will also be the maximum level. Then it decreases. That means if we want to say how the pH, the temperature affects, there is the optimum range beyond which the activity of the enzyme with the pH or with the increase in the temperature will definitely be hampered. Now, the most important part regarding the velocity of the reaction with the substrate concentration. Okay, more the substrate, velocity of the reaction initially increases, reaches to a maximum value which is called Vmax, and after which it is in a plateau stage. That means further it is not decreasing nor increasing. Okay, so these are the we can say the parameters or the factors which affect the enzymatic action. Is it clear to all? After this, apart from these three factors, another factor is there which is called the effect of chemicals, okay, in which we will be doing competitive inhibition, non-competitive inhibition, cofactors, coenzymes. Okay, Amrita, I repeat the substrate concentration. With the increase in the substrate concentration, initially the reaction rate increases, reaches to a maximum value, and this maximum value is designated as Vmax, after which the velocity is stagnant, or we can say it neither increases nor decreases. You can say now it is parallel to the principal axis. Got it? Got it, Amrita? Yes, Amrita, your doubt is clear? Yes, Km is the Michaelis constant. Okay, Shubham. Actually, Km, when the velocity of the reaction reaches to half of the maximum velocity, Km is half of the Vmax. Okay, okay, Shruti, I repeat once again. Let me show this. Vmax is the maximum velocity, Km is the velocity, or we can say when the Vmax or half of the Vmax, that is the Michaelis constant, it is Km. When the velocity of the reaction is half of its maximum velocity, that is called Km, the concentration of the substrate at which the velocity of the reaction is half. Is it clear? Km is that concentration of the substrate at which the velocity is half of the maximum velocity. Okay. Is it clear to all? Okay. Now the next topic. Okay, fine. Okay, yes, I got the feedback. Now let me come to the next topic. It is also the effect of chemicals only, means the enzymatic activity. Okay, students, first of all, here we will discuss the effect of the chemicals on the enzyme activity. Students, two terms are written over here: competitive inhibition and non-competitive inhibition. First of all, tell me, student, what does the word inhibition signifies? And then we will go to competitive inhibition and non-competitive inhibition. What does the term competitive inhibition signifies? What does the term inhibition signifies? Okay, respond. I will see your responses. Okay. 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 Yes, it's mean if we say in one word stopping, in one word stopping, stopping. So the enzymatic activity is also affected by the action of the chemicals. That means the enzymes are very sensitive to the chemicals. Students, how do you know that the enzymes are sensitive to the substrate? or we can say here the chemical also. How do you know that the enzymes are sensitive to the substrate? Through which? Through what? Important mechanism. Can you recall? How do you know that the enzymes are okay, Pragya? Yes, okay. Actually, I want this answer to be very specific. Specific structures. Okay, Pragya. Okay. Students, the answer to this question is very simple. From the properties only we are knowing. Okay. In the previous class regarding the enzyme, when we were discussing the property, and I said you to give me two very important properties or write two very important properties of the enzyme. There you have answered correctly. Give again answer this question. Two properties of enzyme, the important two properties of enzyme. Recall your previous class. Okay. Okay. Accelerate the chemical reaction, and one very important property, yes, catalyst, through which this answer will come, which I have asked you, that enzymes are specific to the substrate. In the property we have studied that the enzymes are highly specific. Yes, very good, Harshdeep. Highly specific in action. What does this mean? What does this mean? If again parallelly I am using the same sentence in other way, that enzyme activity are sensitive to chemicals or the substrate. These are the, we can say, the two different versions of the same statement. Is it clear that enzymes are highly specific? That means a particular type of the enzyme will bind to a particular substrate only, and the chemicals, some of the chemicals will definitely affect the enzyme activity because they are sensitive to it. Why they are sensitive? Because they are highly specific. Are you getting my point, students? Okay. Did you got my point? Okay. Okay, fine. Okay. So here you see, student, there are two mechanisms. One is called competitive inhibition and other is called non-competitive inhibition. In the competitive inhibition, the competitive inhibitor interferes with the active site of the enzyme, so the main substrate cannot bind. Here in this image, blue color structure is the competitive inhibitor, and this is the main substrate, and this competitive inhibitor, it competes with the substrate from binding with the active site of the enzyme because it has structural similarity. Student, can you tell me what do you mean by the active site of the enzyme? See, parallelly I am asking you because we have done all these terms, and it will be again a, we can say, revision for you all. What do you mean by the active site of the enzyme? The site of enzyme action, site where the substrate bind. Yes, correct. The site where the substrate bind. Okay, yes, correct. Now, yes, one example of the competitive inhibitor or inhibition is the malonate resembles succinate, and as a result, it prevents the binding of the main substrate, or we can say the succinate dehydrogenase. So what happened? The substrate malonate resembles succinate. So here this will be malonate, and this will be the main substrate succinate, and this enzyme is succinate dehydrogenase. So succinate will come to bind, but before binding with it, malonate, which resembles succinate, will bind to the active site of the enzyme and prevent the action of the enzyme succinate dehydrogenase. This is a good example of competitive inhibitor. Is it clear to you? Competitive inhibition? Competitive inhibition is clear to you? Fine. And in non-competitive inhibitor, you see the resembling substrate is binding, is going to attach. How? By making a change in the enzyme shape. Okay, by making a change in the enzyme shape. So what I will ask you from this is that, do it bind to the enzyme complex? Do it bind to the active site? Do it bind to the active site? Do it bind to the active site? No. Very good. Very good, Anjali. Very quick response. No, it do not bind to the active site. Rather, it bind to some other site apart from the active site, and it changes the enzyme structure. I repeat, Pragya, the non-competitive inhibitor do not bind. Yes, allosteric site. That is called allosteric site. Very good, Anjali. We were coming to that point also. This site will be the allosteric site, means not the active site, but by binding to this site, the non-competitive inhibitor brings about a structural change in the enzyme, due to which it cannot bind to the main site. Is it okay? Is it clear to you? Okay, fine. Okay. Students, today's assignment I will send you through SNAP homework. By evening, you will check the SNAP homework assignment section, and you will solve and post your solution through SNAP homework only. Today's assignment I will provide you through SNAP homework. So today we will do up to this, and again in our next class we will move to cofactors, coenzymes. Thank you all.